Hello, molecular biology. This is your video update for the week of April 6th. I hope you're all doing well and surviving. Haven't gotten much feedback from anybody, so I assume the way we've been doing things has been working. You'll continue to review the material on your own and listen to the lectures. Again, feel free to send me any messages you need, and I'll work with you and we'll get through this together, okay? So during this next week of lecture, you have a couple of vignettes on some cell biology topics. So there's a lecture on vesicles and protein sorting. So here you just kind of want to get a big picture, realize that the proteins have to be sorted correctly into the correct organelles or membranes where they have to function correctly. So that's pretty important. There's a lecture on the cytoskeleton and microtubules. So this might be a review looking at the different cytoskeletal proteins that are in the cell, what they do, kind of their general role. We'll see things like the microtubules again when we look at the cell cycle and what their role in the cell cycle is. And then the final lecture this week is on stem cells. This is probably where I want you to spend the majority of your time studying, because I really want you to be able to compare and contrast the state of a stem cell, its unique characteristics, to that of just a normal somatic cell. So you want to look at some of the terminology that's associated with stem cells. But also think about it in this idea of global transcriptional regulation, that stem cells are kept in a particular state or transcriptional state, where they can then differentiate into any number of different cells. And this is all done at the DNA level. So how the DNA is controlled, how it's expressed, that enables the stem cell to keep its stem cell-ness, if you will. So once it starts to differentiate, then you start expressing different genes, and you kind of go down this line where it's not easy to go back. But that's where a lot of research is now, is trying to induce the stem cell. So starting from a normal somatic cell and being able to reverse that. If we can understand which genes we have to change their expression, or when we have to change their expression, then we can be able to reverse the clock and be able to make a stem cell out of any normal cell, which would make a lot of therapies easier to do. There are some things that are shared, I guess, between stem cells and cancer cells. Cancer cells are also pretty much able to do whatever they want, and their expression of their genes are out of control, which gives them a lot of unique abilities. So we'll touch on that as well. So those are the topics I want you to look at. Things that are due, there's a smart work chapter that's due this week, I think on the 10th or 11th. So check the smart work system. And then your experimental design. So that's your last section of your grant proposal. You need to look back at your specific aims and see what kind of experiments you need to run. You kind of lay that out. It doesn't have to be in super duper detail, like telling me the ingredients of a PCR or something like that. Excuse me. But you do want to kind of lay out for each aim what you're going to be doing, what kind of experiments you're going to run. Remember, it's more than one experiment per specific aim, so you want to lay that out. Once we're done with that and I give you feedback on that, then you'll be able to put together your whole grant proposal, all the sections, and change anything that you needed to change, and then submit that for the final draft. As for lab, I wanted to mention, once you finish the Western blot activity, then the only thing left to do for the lab is the lab final. So that is a Canvas quiz. It can be on anything we covered in lab this semester. So you want to go back and look at some of the experiments we did, some of the theories behind things like PCR and cloning. And that's going to be due on the 17th, April 17th. So just a heads up that that's the last thing we need to do for lab. It won't open up until the 16th, okay? Okay, take care. Let me know how it's going and stay safe. Bye.